Hi, this is the second video of the brain-computer interface series, and today we'll be talking about the measurement of single-unit activity for the signal acquisition stage of the brain-computer interface algorithm. And if you have not watched my previous video on what a brain-computer algorithm essentially is, I suggest that you look at that video and kind of understand in general what a brain-computer interface essentially does. Now, at the core of every BCI is the ability to detect and interpret neural signals. Neurons communicate via action potentials, and these are essentially just brief electrical pulses that are generated by ion exchange across the cell membrane. And now I'll talk more about action potentials in our upcoming videos, but these signals of action potentials essentially just encode all forms of human thought, intention, and perception. And there are several types of neural signal measurement metrics via measuring action potentials that is used in BCI. And the first method that we're going to look at is single unit activity. Now, this essentially just records action potentials of individual neurons. And the benefit of recording single unit activity is that it offers extremely high spatial and temporal resolutions. Now, the term spatial and temporal resolution in the context of single unit activity refers to the ability to measure and essentially just distinguish the activity of neurons with great precision, both in terms of location and time. And now we can see this by examining this graphical representation, this diagram right here, which is a graphical representation of the stimulus-driven single unit activity from microelectrocardiography. Now, in the production of this diagram, we essentially just examine neural signals in a songbird's brain while providing them with sound stimulus. Here, panel B essentially details the brain regions that were involved in the processing of these sounds, such as the HVC, also used for song learning and production, and other related areas in general. It indicates the recordings were targeted, which potentially focuses on how different brain regions just generally react to sound. And if we look at panel C here, we can see that this is an actual image of a brain with probes that are inserted. The label right here that says depth probe essentially suggests that the recordings are being taken at different brain locations, which just indicates high spatial resolution by pinpointing precise areas involved in neural response to the stimulus. So we can actually see that this idea of measuring single unit activity, providing really high spatial and temporal resolution is actually true. And if we look at panel D right here, it's a sort of frequency analysis. This spectrogram actually shows us that the frequency components of sound over time is recorded in this manner. The vertical axis here shows the frequency, and the horizontal axis here shows the time. This kind of analysis is just crucial for understanding how different frequencies are processed in a bird's brain over time. If we look at E here essentially these are depth recordings, and this panel is just critical in illustrating both high spatial and temporal resolution in the sense that it illustrates spatial recognition because these different colors in the traces represent the recordings from multiple layers or depths within the brain. This idea of measuring it from the surface and depth, which essentially just demonstrates its ability to discern signals originating from different spatial regions within the same brain tissue. And also temporal resolution is high in the sense that the recordings over time, the x-axis ranging from 0 to 4, capture the rapid fluctuations in voltage, which indicates the neuron's responses to stimuli. So each of these spikes or fluctuations here essentially just represent and indicate the neural activity that is related to the processing of that sound, captured with precision in terms of generally just milliseconds. Therefore, the measurement of single unit activity can generally be known to accommodate high temporal and spatial resolution. However, on the downside, measuring single unit activity requires invasive implantation. Now, the measurement of single unit activity just typically uses microwires or microelectrodes that are inserted directly into the brain tissue. And these tools are precise enough to isolate and record electrical activity from single neurons. And here's just generally how the process works. We first start with inserting the microelectrode, and these electrodes are just surgically implanted into the brain area of interest, and these electrodes are just fine enough to position near the individual neurons without just causing significant damage to the surrounding cells. And the second process is just signal amplification recording, where the electrical signals, the spikes generated by the neurons, which are very small, which often just range within the microvolts, these signals are just captured by the electrodes, and then they are amplified so that they can be recorded and analyzed. And then we kind of move into the stage of data analysis, wherein the recorded data that consists of voltage changes over time. This data is essentially used by researchers to just use various signal processing techniques to differentiate between noise and actual neuron spikes. And just kind of how we saw in the last video, how we talked about in the brain-computer interface, there needs to be certain signal processing techniques that needs to be applied in order to process the raw data. Same thing goes on here, wherein we analyze the timings and the patterns of these spikes to understand how the neurons communicate and process information from the brain, but at the same time we differentiate between noise and actual neuron spikes prior to going into that process. Now, compared to other methods of just neural signal measurement, such as those illustrated in this diagram, we can see that measuring single signal activity by using microwires or even microelectrodes can generally be very invasive, which are when compared to alternatives like EEG or even ECOG are definitely much more invasive.